Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Have you recently wondered how social distancing and self-isolation may be affecting your brain? An international research team led by Erin Schuman from the Max Planck Institute for Brain Research discovered a brain molecule that functions as a thermometer for the presence of others in an animal's environment. Zebrafish feel the presence of others via mechanosensation and water movements, which turns the brain hormone on. Varying social conditions can cause long-lasting changes in animal behavior. Social isolation, for instance, can have devastating effects on humans and other animals, including zebrafish. The brain systems that sense the social environment, however, are not well understood. To probe whether neuronal genes respond to dramatic changes in the social environment, graduate student, Lukas Anneser, and colleagues raised zebrafish either alone or with their kin for different periods of time. The scientists used RNA sequencing to measure the expression levels of thousands of neuronal genes. Tracking social density, we found a consistent change in expression for a handful of genes in fish that were raised in social isolation. One of them was parathyroid hormone 2, PTH2, coding for a relatively unknown peptide in the brain. Curiously, PTH2 expression tracked not just the presence of others, but also their density. Surprisingly, when zebrafish were isolated, PTH2 disappeared in the brain, but its expression levels rapidly rose, like a thermometer reading, when other fish were added to the tank, explains Anneser. Thrilled by this discovery, the scientists tested if the effects of isolation could be reversed by putting the previously isolated fish into a social setting. After just 30 minutes swimming with their kin, there was a significant recovery of the PTH2 levels. After 12 hours with kin, the PTH2 levels were indistinguishable from those seen in socially raised animals, says Anneser. This really strong and fast regulation was unexpected and indicated a very tight link between gene expression and the environment. So which sensory modality do the animals use to detect others and drive changes in gene expression? It turned out that the sensory modality that controls PTH2 expression was not vision, taste or smell, but rather mechanosensation. They actually felt the physical movements of the swimming neighboring fish, explains Schuman. Sensing water movements, fish perceive movement, mechanosense, in their immediate vicinity via a sensory organ called the lateral line. To test the role of mechanosensation in driving PTH2 expression, the team ablated the mechanosensitive cells within the fish's lateral line. In previously isolated animals, the ablation of the lateral line cells prevented rescue of a neurohormone that was usually induced by the presence of other fish. Just as we humans are sensitive to touch, zebrafish appear to be specifically tuned to swimming motion of other fish. The scientists saw changes in PTH2 levels caused by water movements that is triggered by conspecifics in the tank. Zebrafish larvae swim in short bouts. We mimicked this water stimulation by programming a motor to create artificial fish movements. Intriguingly, in previously isolated fish, the artificial movements rescued PTH2 levels just like the real neighboring fish, explains Anneser. Our data indicate a surprising role for a relatively unexplored neuropeptide. PTH2 attracts and responds to the population density of an animal's social environment. It is clear that the presence of others can have dramatic consequences on an animal's access to resources and ultimate survival. It is thus likely that this neurohormone will regulate social brain and behavioral networks, concludes Schuman. Story source: materials provided by Max Planck Gesellschaft. Note: content may be edited for style and length. Please support our channel to grow by pressing subscribe button as well as the bell icon for daily science updates. Thank you.